All right, hey you guys. We are starting a new unit today on genetics. Genetics is pretty cool because we get to talk about you, and who doesn't want to talk about themselves, right? Today we're really just going to cover some broad introductory terms that you'll need to know that will carry through the rest of this genetics unit. We're going to end with talking about Punnett squares as well and how to complete those. Some of you will know some of this information already, just having some background information on it. Others, you'll need a little bit of, a recap. So let me just put that up a little bit. All right, so when we look at genetics, we're going to look at some of your traits to begin with. And you can do this on your own or compare your own traits to your siblings or to your parents. Some things you can look at are your earlobes. These are traits that are passed on from parents to offspring. They're either attached or they're free or detached. Thumbs straight up and down, or hitchhiker's thumbs bends back. Over here we have a little butt chin, dimple chin going on. Skin tone, skin and complexion, freckles. In the middle here we have a widow's peak, so your hairline either goes straight across or maybe dips down. If it dips down, that is a widow's peak. Rolling your tongue is also considered a genetic or hereditary trait. The way you fold your hands. So when you fold your hands, if you have your right thumb on top or your left thumb on top, that is something that is genetic or hereditary. This little guy over here has got some cute little dimples and hair. So whether your hair texture, hair volume, straight, curly, all of these, plus way more, are genetic traits that you can look at and compare to your parents or siblings. Some of the terms that we're going to cover include the following. Some of these are past terms that you already know about, others are going to be new. So heredity is simply just the term we use to describe how traits get passed on from parents to offspring, which occurs in your DNA. Genetics is the term we use for studying how this is done through heredity. And chromosomes, which we talked about in DNA, are just those coiled up forms of DNA that you find in your nucleus during cell division. The last two, genes and allele. Genes are the broad trait. Allele is the variation of the trait. So for example, you have a gene for hair color. The allele is the specific hair color you have. You have a gene for eye color. The variation or specific eye color you have is the allele. So gene is very broad, referring to the trait in general. Allele is the specific variation of that trait that you have inherited. Can look something like this as well. So if these are the chromosomes, the light purple, the location, so the gene is flower color. This one is giving off a purple flower allele. This one has a white flower allele. So flower color in general, and then is the gene, specific is the exact color of flower. Same thing over here, you have eye color as the gene. The variations are the alleles. Hair color for the gene. The variations of hair color are your specific alleles. So make sure you understand the difference between allele being the broad trait, gene being the broad trait, and allele being your variation of that trait. Gregor Mendel is the scientist behind all of this. He studied pea plants in the 1850s. He studied pea plants because they were easy to breed, they were fast growing, and had a lot of different traits that you could look at. You could look at height of the plant. They were tall versus short. He could, excuse me, look at the color of pea plant, seed, either yellow or green. He also could look at some seeds were wrinkled, some were round, so a lot of different variations within that one plant. So that's the reason he studied these. Terms that you will need to know very, very well: dominant and recessive. So for each trait you have two or more alleles or letters to represent that trait. If it is a capital letter, that is what we consider the dominant allele. If it's representing a short letter, that is what we refer to as a recessive. So capital letters are going to be your dominant, lowercase letters are going to be your recessive. If there is a capital letter present, that is the dominant trait. That dominant trait always masks the recessive. And this will make more sense when we get into it a little bit here. A few other terms you'll need to know: homozygous versus heterozygous. Homozygous is same, right? Prefix homo means same. Homozygous dominant, then remember that dominant is capital letters. So 2 capital letters are homozygous dominant. 2 lowercase letters are homozygous recessive. A hybrid or heterozygous is simply one of each. One dominant allele, one lowercase. So you'll have to know these terms and how they're described, because when we get into Punnett squares, that's how traits are described. So homozygous dominant, homozygous recessive, hybrids, one of each. Excuse me. Law of segregation. Law of segregation states that when you have these alleles, and again, these alleles are just letters that represent a trait. Let's say it's height in that pea plant. When those plants go to make their gametes, sperm cells and egg cells, those letters are split or segregated out. For example, let's say you have big T, so homozygous dominant. Each gamete only gets one of those letters. So your sperm or egg cells, each of those would only get one capital T. Same here, you have big T, little t, half of your sperm cells would get the big T, half of your sperm cells would get the little t. So of your pair or set of alleles, the gametes being made only get half of that set. They're split apart, they're segregated out when gametes are being formed. Law of independent assortment. This is stating that you have all of these different genes, right? So you have a gene for hair color, gene for height, gene for face shape, dimples, etc. Those genes separate or sorted out independent of each other. So your genes for dimples don't mix with your face shape genes, and your face shape genes don't mix with your height genes. The genes that are specific, or I'm sorry, the alleles specific to that gene stay with that gene. They don't mix around with other ones. So they're independent of each other. Punnett squares. So up until now, we've just going over some just broad terms. Know those terms: homozygous, heterozygous, dominant, recessive. You will also need to know how to complete a Punnett square. It's a big part of genetics. Some of you have already done this in the past, so this will be a review for you. Others, it's brand new. When we're doing Punnett squares, and I'll show you a little practice one next year, you will be asked often to find genotype and phenotype of the offspring. So first, know that when you create a Punnett square, why you create a Punnett square is to show potential offspring results. So when 2 parents are bred or mate, what is the likelihood of their offspring carrying on certain traits from their parents? We look at those traits in 2 ways. Genotype is literally just the letter combination, the possible letter combinations you get in your offspring. The phenotype is the physical trait that those letters represent. So genotype, letter combinations, pheno is the physical trait that is being represented. Let's look at an example. So if this is your Punnett square up here, it is literally a square, a monohybrid cross is called, so you're just looking at one specific trait. So you have a square with 4 boxes. You put the parents' alleles on the outside of the box. In this case, we're using E's. It's always helpful to use letters that you can distinguish easily between capital and lowercase. So E's are a good one to use. In this case, we're looking at long ears. Here we're talking about bunnies, let's say, rabbits. So capital E is representing long ears. It's dominant for long ears. So you have one parent who is homozygous dominant for long ears up here. You have another parent over on the side that's homozygous recessive. So parents go on the outside of the box. The offspring results then go on the inside, and all you do is drag from the top and the side over. I always put, if there's capital letter, I always put the capital letter first because it's easiest to read. So in this case, you have big E from the top, little e from the side. Same over here, big E, from the top, a little e from the side, and then you just keep plugging them in as you go. The results then, so if these two bunnies mated and had offspring, the result of their offspring is that all of them, 100% of them, would have this allele combination, big E, little e. It's the only option present. Also, the phenotype, or physically, what that means is that all of them would have long ears because they all have a capital E. Now, this is not showing that, okay, if they have 4 individuals, all 4 individuals would have that same trait. It's just saying, okay, if every unique offspring that they have, this is the likelihood of that offspring having these results. Again, this will come into play better when we get into some practice tomorrow. This is called a monohybrid cross when we're simply crossing one trait. So in this previous one, we just looked at ears. Long ears versus short ears. We weren't combining ears, length of ears with color of their fur. We just simply looked at one trait. That's called a mono, meaning one, monohybrid cross. A single trait is looked at. Here's one example, another example here. You have pea pod color, you have green, and you have yellow. Green is dominant. Okay, so capital G is representing a dominant color. In this case, it's green. So you have one parent that is homozygous dominant for green. You have another parent who's homozygous recessive for yellow. Parents are on the outside, plug in the letters in the middle. This is your offspring result. 4 out of 4, or 100% of them, are big G, little g for the genotype. And all of them, for phenotype, all of them would be green. Okay, I just want to give you one more example of this before I send you off. Here's just one other example. So here's a Punnett square. I'm using letter B to represent our trait. We're gonna use eye color, let's say. So I'm gonna say capital B is representing brown eyes. It's dominant for brown eyes. Lowercase b is recessive. I'm gonna say one parent is heterozygous for brown eyes, so has brown eyes with capital B, heterozygous, so it has one of each letter, capital and lowercase, and the other parent is homozygous recessive. I then plug in my letter combinations to my Punnett square. These are my results. So I would say that if these two individuals have a child, 50% chance, right here, 50% chance of that child having brown eyes, these big B's, brown eyes, and 50% or half would have the chance of having blue eyes. So again, this is every individual. So these two individuals have one child. That child would have a 50% chance of having brown eyes, a 50% chance of having blue eyes. These same parents decide to have a second child, same proportions would lay out. For that second child would have another 50% chance of having brown eyes, 50% chance of having blue eyes. Often students mistake this and they say, well, okay, if they have 4 kids, then 2 of them would have brown eyes and 2 would have blue. And that's not what we're talking about. It's just for each individual new offspring, this is the likelihood of them having, in this case, brown eyes or blue eyes. The only way to get a recessive trait, in this case blue eyes, would have to have both alleles would have to be lowercase. So I hope that helps you at least start thinking about Punnett squares and these different terms and how they're used. Tomorrow we'll do some practice with this. For now, there is a Quizlet flashcard practice review that I would like you guys to do for your assignment today. So go through those notes again, review those terms, and then complete those questions. All right, thank you. Have a good one.